So, good afternoon, everybody. It's always a great pleasure to have this series of Meet the Expert webinars hosted by the Master in Clinical Science, Clinical Mental Health, in collaboration with the World Psychiatric Association. Today we have two prominent experts, Professor Cloninger and Professor Godwin. Before we go on with the speeches, I will give the floor to Professor Afzal Zaved, who is President of the World Psychiatric Association, to introduce tonight's session. Afzal.
Good evening, good afternoon, dear colleagues, ladies and gentlemen. Thank you very much, Professor Fontoullakis. This is indeed a great series that you have started, not only as a WPA officer, but a great academician and a clinician as well as a researcher. Today's webinar is important as we have got two very prominent scholars who are presenting very interesting topics, which are not only important for our academic or research. We are grateful to them. We are grateful to Professor Fontoullakis for carrying on with these academic activities. And we hope that with the help of his university, World Psychiatric Association will be able to carry on these educational webinars, activities in the near future. Thank you very much, Professor Fontoullakis.
We have with us also Professor Peter Morozov, who is General Secretary of the World Psychiatric Association, and he's going to say a few words also.
Hello, everyone. Thank you very much, Professor Fontoullakis, giving me the floor. I think that this series of the webinars is very important and became very popular among the young psychiatrists around the world. I have a very, very positive feedback, and I do hope that in the very near future we'll have all this series at our website, WPA website, and this important and very stimulating and interesting material appear in our educational portal. We are very grateful to prominent scientists participating in these webinars, and my particular thanks go to Professor Kostas Fontoullakis, who are real engine enthusiast of this important educational program for the young psychiatrists around the world. Thank you.
So we will start with our first guest, a very dear friend. Have worked with him in the past, so it's a great pleasure to have you here, Robert, with us. So we're talking about Professor Robert Cloninger, who will talk about the genetics and psychobiology of personality and well-being. Robert Cloninger is a distinguished life fellow of APA, director of the Arthropedia Institute, and professor emeritus of psychiatry at Washington University in St. Louis. He is widely cited and honored for his biopsychosocial research on personality, psychopathology, and health. He has received the Adolf Meyer, Jude Marmor, and Oscar Pfizer awards from the APA, Lifetime Achievement Awards in Psychiatric Genetics and Addiction Medicine, and is a member of the National Academy of Medicine USA. Dr. Cloninger has published 10 books and over 600 articles in psychiatry, psychology, and genetics. Cloninger is best known for his pioneering research in the genetics and neurobiology of personality and personality disorders. He has developed two widely used tools for measuring personality, the Temperament and Character Inventory, the well-known TCI, and the three-dimensional personality questionnaire. Robert, the floor is yours. Thank you for being here with us tonight.
It is a pleasure to be here and to speak to people in different parts of the world through this mechanism. So I want to thank the speakers, Konstantin, and the World Psychiatric Association for hosting this and giving us the opportunity. I'm going to share my screen now. The topic, as Konstantin mentioned, is genetics and psychobiology of personality and well-being. And this work is based on recent molecular findings in genomic studies of human beings, and as well as in chimpanzees and Neanderthals. And these findings are giving us a deeper and clearer understanding of what personality is and a number of the mechanisms for well-being. I'm going to give you definitions that reflect the new knowledge, introducing these concepts to you, and then I'll give detailed data that will help you understand the basis for these conclusions and findings. Well-being is defined as a complete state of physical, mental, social, and spiritual health. And that can be measured in three ways that turn out to be inseparable. One is physical vitality. A second is functioning with a creative and reliable personality, which I'll define a bit later, as well as with positive emotionality. And the third component is virtuous living, which serves others by moderation, altruism, sublimation, and other virtues. As a result, well-being involves the self-awareness of living as a person who recognizes the dignity of everyone and consequently lets go of fighting, worry, thereby both feels good and does good. Now, let's look at what personality is. You'll see that the definition I gave you for health and well-being is actually very similar to that of the World Health Organization. When we look at personality, that it's important to recognize that personality is not a set of fixed traits. Personality is actually the way people learn to adapt via three major systems of learning and memory. We've been able to show that there are three independently inherited, partially overlapping and dissociable systems that are associative conditioning of our habits and our skills, our intentional self-control of our goals, as well as self-awareness, which is creative and has spiritual aspects that we'll talk about later. It turns out that personality is actually empirically our strongest predictor of both well-being and ill-being. It's important for the full range of mental disorders, for vulnerability to chronic non-communicable diseases, as well as to vulnerability to social dysfunction. And it's also important for health. In particular, human character traits are the components of personality that regulate health by the regulation of the expression of our genes throughout the brain and other organs of the body, which allows us to maintain health and to adapt to changing conditions, to maintain healthy functioning, and to promote resilience and repair of injury, degeneration, and our maladaptive responses to stress. So you see that personality is something that's dynamic and not fixed, and that we actually don't have one system of learning and memory, but three that have to be integrated so that our habits are in accord with our goals and our values, which we perceive in a state of self-awareness. And because our character is something that we create and construct in order to adapt to life in ways that we find satisfying and meaningful, that actually puts us in a position to regulate our vulnerability to disease, as well as to promote our own health and well-being. So that's a general point and a perspective that's leading me to emphasize the importance of the relationship between the person and health, and the importance of taking a person-centered approach to the promotion of health. Now, let's look at the role of precision medicine in health. Precision medicine is an emerging approach for disease treatment and prevention that considers individual variability in genes, environment, and lifestyle for each person. So that's a very important advance in itself because it is person-centered. Its goal is to match treatment to specific phenotypes in subgroups of people in order to predict what's most beneficial. And we're already seeing much work like this in cancer medicine. Notice that this differs from evidence-based medicine, which relies on selecting what works best in randomized controlled trials, which consider the differences in the averages of outcome in different groups. So it's focused on finding average differences between large groups, which are often heterogeneous. And so as a result, the recommendations of evidence-based medicine are not person-centered as precision medicine is. And unfortunately, it also doesn't tell us which particular medicine will work best in a particular individual. So precision medicine is an improvement on evidence-based medicine because it's personalized, but it's still disease-oriented without attention to understanding the processes that promote health and well-being. It's sobering for us as physicians who really want to help our patients to realize that only 16% of health outcomes are attributable to the quality of modern disease care and prevention. And that limited fraction is due to the weak and inconsistent effects of evidence-based medicine and the neglect of health promotion by precision medicine. I wrote about this in a recent article in the World Medical Journal. Now, let's talk about the model of personality that I'm describing to you. This is various referred to as the psychobiological model of personality or the biopsychosocial model of personality. And some people say the biopsycho-spiritual model of personality. It's all of those things, just depending on what you want to emphasize in particular work. But it's measured by the temperament and character inventory so that we can address temperament, which is something involving procedural learning or associative conditioning. And we measure that in terms of traits of harm avoidance, which involves being anxiety prone, novelty seeking, which involves being impulsive and curious about what's new and unfamiliar, reward dependence, which involves our social attachments and our need for approval from others. And finally, a temperament trait called persistence, which keeps us going when things are not going as we hoped or expected, but allows us to persist without giving up hope. In addition to temperament, we measure character traits, which involve our mental constructs of who we are and our goals in life. And this involves how self-directed we are. That is how well we're able to face reality, set realistic goals, and accomplish them. Cooperativeness, which involves our capacity to be kind and generous and helpful to other people. And our self-transcendence, which involves our consideration of things that are important to other people, so that we see ourselves as an inseparable part of something greater than our individual self. And that something greater can be other people, the community or humanity at large, nature, the universe, and some could even say the infinite and the sacred, the divine. So these aspects of character then interact with temperament and regulate the construction of our perception of who we are and what's important to us, what our goals in life are. And as we formulate our identity, that in turn leads us to regulate our temperament and our character in order to accomplish this in an integrated, satisfying, health-promoting fashion. Now, empirically, we've studied the impact of character and temperament on various aspects of well-being, emotional, physical, and social, on all the continents except for Antarctica. And what we see is some data here from studies that I did in Israel with my colleague Ed Zohar. And you can see that these are normalized values. So this line 0 means average. And the people who are low in one or more of the character traits are always below average. And those that are above average are those who are high in one or more of the character traits. And you can see that the people that are high in both self-directedness and cooperativeness are far above average, much more than just being those who are just self-directed, but low in the other two. And you can see that people who are highly self-directed in Israel actually do fairly well in emotional well-being, but not so well in physical and social well-being. So there are things that each aspect of character contributes. And we wanted to understand in a general way, what are the mechanisms in the human genome that have molecular consequences and biological consequences and physiological consequences that then impact various aspects of health. Now, there's another aspect of this that has been more difficult to research, and that is that we've recognized that the healthy and the happy life is also the good life in the sense of the virtuous life, because a universal desire of human beings is that we want health, happiness, love, and to experience coherence with what's good and meaningful beyond our individual self. As I talked about in my book, Feeling Good, The Science of Well-Being, more than a decade ago. And this has been somewhat controversial because many people in modern society are skeptical about virtue. It's not even a word we use very comfortably often. But extensive research now shows that interconnectedness and the inseparability of all living things is not an illusion. And it's not a matter of opinion and belief. Even as biologists, we know that cells are only partially separated by semi-permeable membranes that are actively engaged in exchange of materials in and out of themselves. We also know that all living organisms are aspects of complex adaptive systems in which symbiosis and cooperation are more involved in healthy social and ecological functioning rather than competition and aggression. And at a deeper level, at the most fundamental level, we know that all physical things and all living things are composed of quanta that are constantly arising from and returning to a universal field of energy and information called the Higgs field. So really everything is connected fundamentally, and it's only an illusion of our analytical mind that we think of us as separate individuals who are seeking to maximize what's good for our individual self. And it's also very sobering to remember that no physical model can account for consciousness or for life. And so we have to be more humble in recognizing our relationship to what is beyond human existence. Now, just to stop the general observations and to look at the data about virtue, we've done studies recently. This study was done in Portugal, recently published in the Journal of Positive Psychology. And we used a test called the Virtues in Action Inventory, developed by a positive psychologist. And that's been shown to measure three aspects of virtue: self-control, that is letting go of fighting, being prudent, and caring, which involves serving others, being fair and kind and loving, and inquisitiveness, which is kind of the agnostic view of virtue, because it recognizes that we need or driven to be curious, joyful, energetic, and acknowledges that some people then develop faith in spirituality. But the advocates of this test themselves were rather agnostic skeptics. But what's really interesting is that when we give both the VIA and the TCI to people in large populations, we find that self-transcendence is consistently related to virtues, as is self-directedness and cooperativeness too. And also persistence is consistently related. So what we've discovered recently is that we can understand this by looking back at some of the ancient wisdom literature, particularly Plato. And if we look at his metaphor of the charioteer, in which we have a charioteer who's guided by an unruly black horse and a noble white horse, that we have an understanding that the charioteer represents reason, which is intuitive insight inspired by virtue. And this is what we call the creative character in the temperament and character inventory. That is, individuals who are high in all three of the character dimensions. And the unruly black horse represents our appetites. That is, our rational emotional drives, like being harm avoidant or anxiety prone, being impulsive, risk-taking, and needing approval from others, looking for things, in other words, outside of ourselves in the external physical world. And then the noble white horse is actually the horse that listens to reason and helps the charioteer to reach his goals with courage and perseverance or resilience. And this is measured by the high persistence, as well as high self-directedness and low harm avoidance, so that people are determined, goal-directed, and not frightened easily. And it's the interaction of these three groups of forces that help us to make our goal, our habits, to be in accord with our values and our vision of who we are, what our identity is. Now, what that means is that we go through different stages of awareness as we mature and integrate ourselves. We may begin as unaware creatures who learn by behavioral conditioning alone. And these are people that I describe as being emotional and unreliable. We can progress by using our analytical mind to learn by intention, to be self-controlling intentionally, that allows us to regulate our habits and to set individual goals for ourselves. These are individuals who are high in both self-directedness and cooperativeness to the degree at least of seeking mutual benefit. They may not help you if it's going to hurt them, but they will help you if it's mutually profitable. That's egocentrism. And then we can progress from there to allocentric thinking, like a good parent or a teacher or a coach. And here we are beginning to use self-awareness to integrate all three of the learning systems that we have. But we're still relying mainly on our physical senses and our analytical mind, which uses uncertain assumptions about values. And these people we could describe as in the early phases of being creative and reliable, or post-materialists. And then finally, we have a level that's achieved in rare individuals, but more and more common, especially in younger generations, of people who are more contemplative and directly perceive what's truly valuable for themselves and others. And their intelligence are all well integrated to bring habit in accord with goals and values. And these are full-blown cultural creatives. Now, we can look at this in terms of another one of Plato's metaphors or allegories, and that's the allegory of the cave, in which the unaware people are really prisoners trapped by their irrational desires, only looking at shadows on a wall in front of them that are themselves projected by people who are pulling their strings or fooling them for their own advantage. And the self-serving image makers who are casting the shadows that entrap a gullible people are using a technology to cast illusions by shadows and propaganda. And I'll let you reflect on to what extent this is happening in contemporary society. And then the allocentric individuals are those who are starting to climb out of the cave. They've stopped manipulating the prisoners and they turn around and they start to climb out because they get glimpses of sunlight at the opening of the cave and they seek to reach the source of that wisdom, that light. And then finally, we have people who are in a state of contemplation and highly self-aware and well integrated, who are lovers of wisdom, who are out of the cave and illuminated by the sunlight, although they may go back into the cave to help the others. So this is a powerful metaphor that lets you know that personality is something that's dynamic and can be cultivated. And we're going to examine some of its molecular mechanisms now. I'm going to give you first a summary of what we've discovered about the three networks for learning and memory in modern humans, in addition to general intelligence. Because we've actually identified almost 1,000 genes that fully account for the heritability of personality. And as a result of that, largely account for the heritability of well-being. And these systems we've been able to identify based on studies of the genomes of modern humans, as well as those of chimpanzees and Neanderthals. And that's published in Molecular Psychiatry. Now, these three networks have emerged and evolved in response to adaptive challenges over the long periods of evolutionary history for modern humans. And associative conditioning, as we know it in modern humans, is actually nearly fully developed in primates, early primates, 41 million years ago. And we can recognize this in the people who are highly emotional, who don't have much character development, and are responding primarily to their temperamental-driven drives. We describe the genetic network for that as the emotional reactivity network, which operates by behavioral conditioning or habitual responses to extracellular stimuli. And we've been able to identify that the genes that make this up, there are 249 of them, are involved in the two networks that allow associative conditioning to respond to extracellular stimuli. This is the ERK and the phosphatidylinositol 3K system. The second system that evolved a little less than 2 million years ago in working man, or Homo ergaster, the ancestor of Homo erectus and the common ancestor of both Neanderthals and modern humans. These individuals developed intentional self-control in order to seek food, to make tools, and other goals. And we've been able to show that this system of 438 genes operates based on regulation of the phosphatidyl phosphoinositol calcium second messenger signaling system within cells. The third system emerged 150 to 100,000 years ago in modern humans. And this is the network for the creative prototype that genetically we've called the self-awareness network. And we've been able to show that that maps directly onto the default mode network and the autobiographical memory system that is to integrate gene expression and to regulate gene expression in order to be able to maintain allostasis, that is, maintain health under changing contexts. And in our evolutionary studies comparing the genome of modern humans to that of chimpanzees and Neanderthals, we were able to show that what's unique to modern humans are long non-coding RNA genes that allow us to coordinate groups of genes and cells to adapt to our internal perceptual models that by which we integrate our desires, habits, emotions, ideas, and values as a whole. And that's a fairly complicated system of 574 genes that occur in clusters of genes in which there's at least one gene unique to modern humans. So that's the general findings that we have. And I'll go through the data for that so that you understand the basis for it. We did 3 independent studies, one in Finland, one in South Korea, another in Germany, with 2 to 1,000 subjects. And we used the TCI in all of those subjects. And we used various platforms that had at least 1 million SNPs that we measured. And these are in a general longitudinal birth cohort study, in a sample of healthy twin families in Korea. And we looked at only the unrelated subjects in that. And we also had healthy people that had been identified in Germany, excluding all personality disorders and any mental disorder, at least to the best that they could. Actually, the selection was probably only very effective for excluding people with antisocial personality disorder. And that turned out to be very interesting because it showed that when you select against antisocial personality disorder, you select against the personality types that we've described as being associated with personality, and hence the genes for that. In any case, what I can summarize from those findings is that we found 3 nearly separate networks of people with different combinations of temperament and character. We found that the personality dimensions don't occur independently of one another, but they tend to occur both at a phenotypic level and at a genotypic level in clusters, where we have the creative and reliable cluster, the organized and reliable cluster, and the emotional unreliable cluster. And you probably didn't notice it in the earlier slide because I went so fast, but these clusters have also been recognized in the World Value Study. And this is what's called the traditional cluster. These are the modern materialists, and these are the cultural creatives, or the post-materialists. And there's a lot of complexity within each network that we can explain with the temperament character relationships that are within it. But these are very interesting, powerful groups of people. In addition, we found that there are 3 nearly separate genetic networks that correspond one-to-one strongly with the phenotypic networks. And it turns out that 67% of the genes in these networks are unique to only one of the networks. So even at a genetic level, these networks are highly separated. But in addition, we found that environmental factors also distinguish the networks in a one-to-one basis. And we were able to show, for example, that the creative individuals are those who tended to be reared in conditions where there was a lot of parental warmth and tolerance, whereas the unreliable and emotionally reactive individuals were raised under a lot more conditions that were intolerant and fear-driven. Now, as you might guess, since these are genes for personality, they nearly all are expressed in the brain. In fact, they all are. But they're 70% of them or more are also expressed in nearly all the organs of the body. And these shows all the individual organs, but that captures the heart, the gut, the endocrine glands, and the influences on the hypothalamic pituitary adrenal axis. And so we see this throughout the body, and that positions these same genes, which are overexpressed throughout the body, to regulate not only personality, but the assist in the regulation of health by our personality as we learn to adapt to changing environmental conditions. And we're now doing studies with the actual transcriptome to show just exactly what those mechanisms are. And what's essentially happening is when we activate our awareness of who we are, what we desire, what our relationships to others, and what makes life meaningful, we promote mechanisms for healthy longevity, such as increasing heart rate variability and the molecular processes for energy production, immune response, healing of injury and stress, and neuroprotection. We also promote prosocial and virtuous behaviors, like by virtue of identifying with others, and moderation and ecological intelligence to protect the environment, because we develop an outlook of unity with other people and nature. And it also promotes creative and innovative actions via the activation of the late myelinating and recently evolved regions of the human brain that are unique to modern humans, which includes not only the late myelinating neocortex, but also the neocerebellar cortex, which has actually evolved more prominently in modern humans than it was in Neanderthals. The cerebral neocortex and the cerebellar neocortex have co-evolved. Now, when we look at awareness, we not only see that there are these mechanisms that promote all the aspects of health and well-being, but that our self-awareness can be either enhanced or blocked depending on environmental conditions. It's enhanced by promoting creativity and narrative thinking, and things that promote generosity and charity, and by mindfulness and other forms of contemplation. On the other hand, it can be blocked by impairing any of the components of persistence and resilience, such as promoting fear, which increases harm avoidance, or emphasis on custom and authoritarian control, which decreases our self-directedness, or by allowing social inequity and elitism to decrease our hope, and makes us feel more helpless and decreases our persistence. So these are the mechanisms by which we can understand how the same set of genes for character can influence how well we're able to regulate our temperament and how well we're able to realize our goals and our values. Now, we've been able to bring this into studies of evolution of individuals, of our extinct ancestors, the Neanderthals, because of the fact that the relationship between the phenotypic networks, that is what we can measure with the temperament and character inventory, is extremely strong with the genotypic networks. And this size of this and the strength is shown by these circles. And anything off that is actually quite weak. This is the weak relationship, and the green is the strong relationship. So essentially, we're able to observe the personality of Neanderthals by just looking at their human genome. They are humans, and they're, as you know, their protein coding genes are very much like ours, as is true for the chimpanzees. But there's been a change in the non-coding DNA of humans. We looked at the standard genome of Neanderthals and chimpanzees, and we compared it to the Homo sapiens genome. And we found that of the 972 genes for personality that are associated with learning and personality in modern humans, chimpanzees have 553 of them, and Neanderthals have 652. Now, if we look at these three species, the chimpanzees, the Neanderthals, and the Homo sapiens, and we also look at the three learning systems, the extent to which they have the genes in those, you can see that the red refers to the non-RNAs, and the coding RNAs are those in blue. And so you can see that the chimpanzees basically have only the protein coding genes and a small number of non-coding, small, short non-coding RNAs. And if you look at the Neanderthals, they've got a moderate number of long non-coding RNAs. And those are true in all three of the systems. But the Homo sapiens has a big increase in long non-coding RNAs, larger than that in the Neanderthals. Now, if we just look at the genes that are unique to sapiens by gene type, you can see that there are only 5% that are coding RNAs. And the big increase is in the long non-coding RNAs. There's also an increase in pseudogenes, which also influence regulation of personality and cognition. So most of these non-coding RNAs in sapiens were long non-coding RNAs that can evolve very quickly and whose function is to regulate the expression of genes and also the co-expression of genes in clusters, so that they can then take over the regulation of protein coding genes and the epigenetic processing of those genes, which allows us to regulate allostasis epigenetically. That is, you can, by increasing awareness and learning how to live in a way that is more health promoting, actually transform the expression of your own genes. Now, in a little bit more detail, 95% of the 272 genes that we found unique to modern humans are not protein coding, and nearly half of those are long coding RNA genes. There were some of these already present in Neanderthals, 14%, but modern humans have more, 21%. Let's see if I can go back. And what we also found was that the promoters of the link RNAs are more often under positive selection than their exons, so that we know that these are carrying out a regulatory function. What's particularly interesting is that the link RNAs unique to modern humans and under positive selection are functionally different from those found in Neanderthals. 70% of them were found exclusively in the genotypic network for self-awareness. And these are the genes that are associated with the expression of creativity, longevity, and health when people are reared under conditions of parental warmth and tolerance, which leads to an outlook of unity and connectedness. So genes and environmental factors can shape our formation and expression of these genes, and we can take over control of this consciously as a result of modern human self-awareness. So there are other things that are unique about modern humans, and one of them is our capacity for extended longevity after reproductive age. Most animals after reproduction don't live very long, but modern humans are exceptions in that. Healthy longevity provides the opportunity for altruistic grandparents to help their children and grandparents to be able to survive, reproduce, and flourish. So healthy longevity combined with a capacity for altruism has allowed for positive selection of long non-coding RNAs, which have allowed us then to develop healthy societies at times that are not as divided as we are at the moment in the world, and where strangers and other people try to help one another rather than to cause one another problems. Now, this was something that the Neanderthals did not do very well. They tended to live in small groups that became highly inbred, and they may have contributed to their own demise. But certainly we had a greater fitness capacity than the Neanderthals. So you can see that the summary that I gave you initially, we've been able to work out the way to measure these traits. We've been able to identify the gene sets that influence that, what the mechanisms are at both a behavioral and cognitive level and at a molecular level. And so we're in a position then to think about the implications that this has for us as physicians who want to promote health and well-being and to reduce disease and suffering. So we have to then see that human health is more than the absence of disease. We evolved and became successful as a species because of our increased capacity to develop a state of well-being, to regulate allostasis. And that state of well-being applies to all three aspects of the person: our body, our analytical mind for intentional self-control, and what we can call, I think, without any embarrassment empirically, the soul, which is something that we recognize through our self-aware mind. Now, this ternary structure of human beings is empirically grounded in what we've learned about the structure, development, and evolution of human personality. And then human personality profiles, we know, are major risk factors for most mental disorders, for non-communicable diseases, and mortality and spiritual dysfunction. If you look at a list of the actual causes of morbidity and mortality, it's consistently personality-related behaviors like drinking and unprotected sex and violence and so on. The development of well-being, we can also see, depends on our capacity to integrate the three major systems of learning and memory that have evolved in us. So effective health promotion is a creative process that depends on simultaneously promoting plasticity, virtue, and creative functioning. But we've got to be able to change. We've got to want to know what it is we want to become. And we have to figure out the ways to implement that are a creative process. So if the most effective healthcare requires a person-centered approach, and it cannot be reduced to somatic and pharmacological approaches alone that do not involve learning about how to be healthy, compassionate, and creative human beings. But remember too, that somatic and pharmacological approaches are very useful tools in stabilizing patients to engage effectively in a more comprehensive person-centered approach. But alone, they often lead to chronic disease management without cure or good health, or at least not without optimal health. So I'll stop there and just remind you, if you don't remember it, this is the evolutionary picture of our ancestors, where we go from the Australopithecines, the Homo habilis, to Homo ergaster, the working man, who then led to both Homo erectus and Homo heidelbergensis, who's the most likely common ancestor of both Homo sapiens and Neanderthals. And there are increasing levels of well-being on this axis, as well as numbers of years decreasing. Here are some references for you. There's a link that will take you to the most recent articles that all have links to the others. So I'll stop there, and thank you for your attention.
Dear Robert, I would dare to say that this is one of the most intriguing lectures I have ever attended. It was exceptional and well within modern questions and philosophical wanderings, who we are and where we're going. And it verifies an impression I have that personality characteristics are very few. There are not the dozens. Many authors suggest there are very few, but they are under a complex regulatory system, which modifies their expression, but the core traits are limited, maybe 3 or something like that. Is that what you think?
Yes, because we can see it, that our whole personality is based on these 3 systems of learning and memory. And so the whole idea of there being many separate traits is a bit of a fallacy, because the traits themselves are nothing fixed. They're something that emerge in the process of our adapting. And the core traits are these 3 systems of learning and memory, which we recognize as a profile of traits. And even at the genetic level, those profiles are what is inherited, not genes for individual components.
I have seen in our research, we have seen some radical changes with age, radical changes in the way these 3 systems, we have different modules seen, but in more or less they correspond to what you showed. So what we showed was a chiasm of these traits, maybe at the age of between the age of 25 and 30, that more stability is developed, less compulsivity, more reason, less emotionally explosive temperaments. Do you think there is something like this in the aging, in the maturation process throughout the lifespan?
Yeah, there are several processes involved, of course, because you have the maturation of the self-awareness system, and you have just practical exposure to feedback that can lead to modification of habits. But it very much depends on the quality of the environment and what we're encouraged to do. And so if you are living in fear and violence, then there's less opportunity to grow in self-awareness. And so you can have both health promoting processes that increase or health compromising processes that increase. Because as we age, at a certain point, we become less impulsive, but then later we also may, in fact, we've shown that people become less innovative and creative. And then later they become they have less plasticity and greater fragility. And so the study of this over the lifespan is really, I think, very promising. And we have the opportunity to understand the conditions to get at the most fundamental processes, because when we don't treat the core cause, then we never really make much progress. And so now I think we can get more at the core learning processes and also then try to create the conditions for their expression, because what you see with aging is epigenetic change.
Let me make you a final question from my side, because we have one from the audience. Since you showed the evolutionary tree with Neanderthal genes and so on, the average survival age for humans back in the Pleistocene was something like 25 to 30 years. This was the period during which these personality traits were hardwired in our genome and in our body in general. So, and this survival age coincides with this shift in brain and psychological function. Are surviving people over the age of 30 or 40 against their evolutionary programming?
Well, no, actually what happens is that the increase in longevity is a characteristic of Homo sapiens. And the adaptation occurred when the common ancestor of Neanderthals and sapiens spread throughout Europe. Then we had the Neanderthals in Europe and Asia under different conditions than the people in Africa. And Africa turned out to be a more hostile place that was more challenging, with less predictable food supply. The Neanderthals could just move from one place to another when their resources were limited. But our human ancestors in Africa were faced with extinction unless they became more creative. And that seems to be the critical difference in the stress. So the striking climatic changes that were harsher in Africa at the time than in Europe actually prepared us as a species to be more cooperative, to live longer lives, to have greater resilience, to be more have greater capacity for allostasis.
Yes,
and that's why it's connected.
This explains the differences between Neanderthals and sapiens. What I asked is that it seems that the average survival age for sapiens was 25 to 30 during the Pleistocene. And what we see in modern humans is that at exactly that age, from 25 to 30, we have a radical change in personality, a maturation stage.
Oh, I see what you're saying.
Does modern middle-aged humans go against their evolutionary programming? I mean, the natural state of human personality is the young personality we consider today immature, but this is the more.
Well, what we
just
see,
a comment. All these are hypothetical.
What we see in longitudinal studies is that the growth and change in personality does tend to level out after age 30. It's already starting to peak at 25, but between 25 and 35, it's definitely peaking. And it's at that point that self-transcendence is pushed down in modern secular societies. And so our capacity to continue changing is impaired just when the self-awareness system seems to be fairly mature. And yet I find that it's possible to stimulate this at any age. I don't think we lose the capacity. I think that it just, there's less plasticity. It requires more understanding to give just the right combination of help and encouragement to get people to grow at an older age. But it's definitely harder to get people to change the older they get, on average.
So there is one question by Dr. Arnab Datta, who asks, our current disorders are primarily based on impairment in productivity. How does productivity and socio-occupational independence figure in this relationship between social virtues, the awareness network, and well-being?
Would you say the first part again? I'm sorry.
Our current disorders, mental disorders probably, are primarily based on impairment in productivity. So the question concerns how that, productivity is related to social virtues and all these.
Well, that's an important question. Thank you for it too. What we see is that people who are not happy, who don't have a sense of purpose and meaning, don't have the same persistence, plasticity, or creativity in their work. And the virtues really are a way of remaining flexible and adaptive, not only thinking of your individual self, but your contribution to your corporation, your family, your community. So people who are virtuous are the basis for healthy communities and healthy workplaces. And I think we see with the modern problems with burnout in medicine and all employees really, that as a strictly profit-driven business model has taken over everywhere, that more and more we see burnout, which is I see as a demoralization and a loss of people's ability to be who they feel they are, to express that in their work. So it's really frightening in medicine to see that 60% of doctors would not encourage their children to go into medicine, and 20% now who are still in it want to quit in a few years. So we can see that we need to actually have more person-centered approaches to the workplace and to the community and so on in order to decrease burnout and to increase profitability, but not just profit for its own sake, because that only benefits a few, and it leads to resentment and conflict and division and distrust, and all the things that we see so prominently around the world now.
Afzal, Peter, do you have anything?
Yeah, well, thank you very much, Professor Panger. This was really an interesting talk, especially talking about very difficult aspects of personality disorders, and well-being. Now, as we are having many innovations in the field of genetics, and you have in a very interesting way explained the genetics and the personality and the well-being. Do you think that there is a chance that genetics will help us in understanding the treatment or management of personality disorder in coming years?
I do. I think that we already see in what I presented implications for how we need to approach patients. What I don't think is helpful is to talk about the disease risks associated with individual genes. You see that the genes really are working in groups and in clusters, and they're regulating adaptive functions. And so, although we might want to be able to repair rare abnormalities, I think the idea that we can find just a few genes that to target is probably not going to be as profitable as understanding how to promote the regulation of our genes. And so, for example, when 23andMe gives us a profile of what you're at risk for, I would say, rather than fight the disease, why not help people understand how they need to live their life so that that vulnerability won't be expressed? And because we have the adaptive capacities that have helped us to adapt, even though we carry a lot of disease-prone genes. And I think that's really what we need to understand, that it's a mistake to focus too much on a discrete disease, because the diseases turn out to interact with one another. We see that over and over whenever we look at what does, at a community, at a public health level, what do we have to do in order to reduce Alzheimer's? What do we have to do to deal with diabetes and cancer risk? Yes, we can intervene when someone's already sick, but why don't we do more work to promote healthy public health?
Peter?
Well, thank you very much. Really, it is very important for us, particularly for clinical psychiatrists, because personality is one of the basic stone when we make a diagnosis or make a prognosis or approach to treatment. My question is more general, probably a little bit naive. From evolutionary point of view, what is the how it's a proportion in the influence of the concrete personality from genetic factors, 972 genes, which could be followed from chimpanzee, from Neanderthal man, and epigenetic, which is there are a lot of influence on the genes. They are it's a permanent process, and if they are really, because if we would like to influence on gene, so it's mean we should use some epigenetic factors in any case. Well, what is the proportion?
Well, the heritability of personality is about 50%.
Mm-hmm.
And the influence, but you should remember that we're not limited by what we can do by that 50%, because these are learning systems. So both genetic and environmental factors are important. You can never have one without the other. And so the whole idea of, is it a strong heritability or weak heritability, really is not decisive. But the fact is that our capacity for thought and awareness is manifest on the basis of these molecular processes in our physical body and our brain. But then we become aware of what we value, what we want to work toward. And that is what is the most powerful epigenetic tool. I don't think we have to use medications to do that, although, we may learn how to do that better with the aid of medications.
Mm-hmm.
And even what we're going to hear from Dr. Goodwin, about use of psychedelics. When someone is depressed and older in age, can we give them a powerful stimulus with psychedelics to reactivate some aspect of their awareness? Or do we do it only with the process that mystics have used and sages have used throughout time? I think as a clinician, you don't have to measure the genes in order to know that they're working, but you need to understand how they're working. And that's what I think this is most relevant for the clinician, because you can just look at the personality profile and know what's going on underneath that.
I see.
But then how do you work with someone who has no interest, for example, in changing their life? When I work with patients and when I work with drug addicts, I often ask them, well, what gives you, what makes you feel good? And they'll say, well, cocaine or opiates. They may remember that they've had loved ones and so on, but now they turn their back on it. How do we refocus them on what's valuable? We can't do that part right now. We can't give someone the motivation to want to change.
Thank you.
We can discuss things like this eternally until next year. We need to go to our. Thank you very much, Robert.
My pleasure.
As said, this was one of the most intriguing lectures I have ever attended.
Well, I'm going to stay and listen to Guy.
We will have a lot of things to discuss after Guy's talk, I think. Now, Professor Guy Godwin will talk about psychedelic drugs and psychiatry. Is back to the future with a question mark, with a big question mark, I presume. Guy Godwin is CMO of Compass Pathways, Emeritus Professor of Psychiatry at the University of Oxford in the UK. He completed his medical degree in physiology at the University of Oxford, and following training in psychiatry, became a clinical scientist and consultant psychiatrist at the MSc Brain Metabolism Unit at the Royal Edinburgh Hospital in Edinburgh, UK. His research interests have been in the treatment of bipolar disorder and the application of neuroscience in understanding the neurobiology of mood disorders, with a focus in developing new treatments. His current interest is the potential to transform treatment using new technology and new drugs, notably the psychedelics. He is a fellow of the American College of Neuropsychopharmacology and has previously held the position of president of the British Association for Psychopharmacology during the years 2004-2005. He is past president of the European College of Neuropsychopharmacology and emeritus senior investigator on the faculty of UK National Institute of Health Research. He is a Thomson writer, highly cited researcher, classified at the top 1% in the field. Guy, the floor is yours.
You can see these slides, and this is the title, Psychedelic Drugs in Psychiatry, Back to the Future. So just to start with disclosures, I'm currently employed, as Kostas said, by a company, Compass Pathways, that is seeking to really restore psychedelics to some sort of place in medicine. So I have that very obvious, I don't know, it's a conflict of interest, but it's a clear interest. And in addition, I've consulted with a number of companies of various kinds in the last year or three. What I'd like to do this afternoon, this morning, this evening, wherever you are, is to give you a brief history of these medications, which are often called hallucinogens, but we need to be a little careful about terminology, I think, and we'll be a bit more specific in defining where they act, etc. I want to give a little history of the original medical era, because there was one, and it basically went wrong for interesting reasons. And we're now in a situation where we're trying to go back to reinvent an approach that we really lost for 2 generations. And it's not yet clear that we're going to be successful. So we have no reason to be complacent about the present situation, but it's a very interesting and fluid one. So the history of psychedelics, hallucinogens in the West really started with the discovery of LSD. And this is probably well known, was an accidental discovery. It was made by a Swiss scientist, Albert Hofmann, who worked as a chemist at Sandoz in Switzerland. And in 1938, he was involved in synthesizing a number of compounds derived from ergot. One of them was LSD. He was interested in applications really more to do with the peripheral nervous system, blood pressure, etc., than anything to do with the central nervous system. But for some reason, which he himself can't explain, he had a hunch about this chemical entity, this LSD, that made him return to it in 1943, which was fully 5 years later. And he had a slight intuition that this was going to be an interesting drug, and he eventually administered what he thought was a low dose of the drug to himself to discover what it did. This was not at all unusual, really, until the present day, although I think probably much less now, that the inventors of chemicals, the inventors of drugs, would actually try them out on themselves as a sort of first step in the process of making them medically useful entities. So his first chosen dose, was 250 micrograms that he thought was a very small dose. And that dose had a very dramatic effect. And, I'm not doing well this. He gave the count, what actually happened was that he took the drug and then decided to cycle home for lunch. And on that bicycle ride, he started to have an increasingly strange experience. And if you like, the most obvious thing that happened was that he had very striking changes in his visual perception. So that, as you see here, he began to enjoy unprecedented colors and plays of shapes that persisted behind closed eyes. So kaleidoscopic, fantastic images, alternating, variegated, opening and then closing themselves in circles, colored fountains, constant flux. So this is just part of the experience. In addition, he began to feel extremely anxious and paranoid, very frightened. So we know from the beginning, and this, remember, was someone who was completely unprepared for this experience, and preparation turns out to be very crucial in how we're going to use these medicines in the future. He had this really extraordinary experience, which had a number of implications. The first was that this was a clear change in how people perceived and experienced the world, which was produced by a drug, and a very small dose of a drug. And at the time, remember, 1943, there was really no clear understanding that neurons communicated chemically, that the brain was essentially a chemical machine. And the leading authorities at the time, someone like Jack Eccles, who was the leading neurophysiologist, actually believed that synapses were electrical devices. And so it was only about 4 years later that the rest of the world caught up with what was really obvious from Hoffman's experience, which was the highest functions of the brain are mediated as a chemical machine. Now, what was the chemical? It's shown on the right, that was LSD, and on the left is shown the analogous structure that we now call serotonin, or 5-HT, 5-hydroxytryptamine. And 5-HT had barely been discovered in 1943. So it was initially established to be exist in the gut, where most serotonin actually still is, and only later was it really discovered and proven to be in the brain. So it turns out that what was happening was that LSD was activating receptors for serotonin in the brain. And we now know that these drugs are active at a particular receptor, the 5-HT2A receptor in the central nervous system. Now, there's not a completely exclusive effect, but it is widely believed on the basis of both animal experiments and also some human experiments that this is the key receptor for the effects that are most characteristic of these drugs listed on the left, that is LSD, DMT, 5-methoxy-DMT, psilocybin and mescaline. Now, there are a large number of drugs of this type, and they occur naturally, with the exception of LSD, in a number of different preparations, some of which are associated with the practices of particularly indigenous peoples across the world, but particularly famously in South America, and the use of particularly of ayahuasca, of toad venom, and of the other psychedelics, as we now call them, was discovered and, in a sense, publicized in the 1950s, but had been described before. So these drugs were not new to humanity, but they were certainly new to the West as synthetic elements, and of course they were our understanding and the base of science has developed on the basis of what we have done. There are, in addition, drugs, dissociative drugs, that are active at NMDA receptors. And I mention them because they have also, they probably are part of the mechanism of action of some of these traditionally used compounds. And so the dissociative drugs act apparently at NMDA receptors, and the particular compounds that we know from Western pharmacology are ketamine, phencyclidine, and indeed nitrous oxide, which seems to work at these receptors. Now, I'm assuming that most of you are psychiatrists, and you'll be aware that interest in these drugs in treating depression as rapid-acting medicines is also very current, and is part of this, I think, very exciting new wave of treatments that we're having can become available in the last decade, particularly for depression, which I'll talk about. But let's just pursue the history of LSD. LSD was, as you've heard, made by Sandoz, and in those days, regulation was, to put it mildly, very light touch. So it was made available to particularly psychiatrists because it seemed obvious to the people who took it and observing the people who took it, that it had effects on the mind, that it might have potential for psychiatry. And so it was made available to interested psychiatrists, basically for experimentation on their patients. And as you can see here, this little booklet, this is the package insert, if you like, which was only 4 pages, if you actually take a look at it, really describes in the following terms its use, which is for analytical psychotherapy to elicit release of repressed material and to provide mental relaxation, particularly in anxiety states and obsessional neuroses. So that was what the initial use of these medicines indicated it might be helpful for. The average initial dose was 25 micrograms, which is, as you may recall, 1/10 of what the large dose that Hoffman took initially. And the recommendation was to use this repeatedly at psychotherapy sessions and in increasing doses until some ideal optimum was reached. And the statement here, as you can see, is that in individual cases, very large doses may be necessary. The recommendation is that this should be once a week. And in addition, it's noted that this can be done as group therapy. The average number of treatments required varies from 7 to 10 or 14 to 15 in more severe cases. And there have been as many as 40 cases. So this is therefore the emphasis, and it's noted that proper psychiatric supervision is essential. So there we are. That's the start of the story with LSD. Its use as described there was characterized as being used as a psycholytic. So it was thought of as mind loosening. These relatively low doses were seen as an aid to talking therapy, and it was associated with particular individuals who both used it and promoted it as a useful way of helping patients. Now, bear in mind that this is at a time long before DSM diagnosis, when most psychiatry that didn't concern psychotic patients really dealt with the concept of neurosis. And the concept of neurosis was less of an activity of diagnosis than a way of providing an explanation about patient symptoms that was based very heavily on psychodynamic theory. Famously, Cary Grant had 60 sessions, and it was noted by Michael Pollan, who wrote quite graphically about this era in his book, How to Change Your Mind, that it was an excellent business model because people would keep coming back, perhaps for the drug, perhaps for the therapy. But anyway, they mainly paid for the therapy. Now, what is sort of lost in history, but has been looked at again by people who have gone back to the literature that developed at the time, is that the best outcomes were really described for anxiety, depression, and obsessive compulsive disorder. And the broad ballpark numbers that came out for good outcomes were around between 40 and 70%. Now, it's very difficult to map those reports onto what we now regard as adequate practice for RCTs. So there was very little sense that one needed to randomize patients to one treatment or another, very little sense that you necessarily need to use placebo, that you might compare different doses. There was just a rather poor quality literature. It's really been reviewed quite well recently by James Rucker, and it's certainly worth looking at that if you're interested. Now, the second way in which LSD was used was as a high dose and truly psychedelic. And the word psychedelic was coined by a man called Humphrey Osmond, who was a British psychiatrist who developed, working mainly in Canada, who developed an early interest in the effects of high doses of these drugs. And those doses produced much more profound and absorbing experiences that are not really amenable to interactions with a therapist. So they are essentially personal, inwardly directed, and can be very dramatic and are described as such, as a transcendent experience. And the objective, the very explicit objective of this, was to obtain a kind of spiritual epiphany. And the most famous person who kind of took this dose and wrote about it was Aldous Huxley. And Aldous Huxley was a remarkably good writer in English, and the book is well worth reading now because it's mercifully short, but he wrote a book called The Doors of Perception. And he became very interested in it and saw it very much as linking to his sense of spirituality. So I think one of the difficulties with this approach was that it was inherently demedicalizing. So it kind of took people out of, if you like, what was then the psychiatric paradigm of psychoanalysis, and it moved you into something that was much closer to religious experience and breakthrough experiences, mystical experiences, which of course very attractive at one level, but were not essentially they weren't the terrain really necessarily of psychiatrists, even though psychiatrists were interested and promoted this approach. The medical use was not enormous, but it resulted in quite a lot of publications. So this was by no means a secret, and there were about, it's estimated, 40,000 patients were treated in one way and another. Now, what also happened was that this became a kind of publicly known fact that these drugs had dramatic effects on perception, consciousness, and the understanding of the world. And that sort of became bound up, this recreational use started outside of medical control, which in a sense became as its object the transformation of society. And the person most associated with that was Timothy Leary, and he is often credited to have been the leader of the psychedelic movement, as it was often called. And he asked interesting questions like, is there a life after youth, as you can see on this slide, to which the answer is obviously yes, for those of us on this panel anyway. So Timothy Leary coined the phrase, tune in, turn on, drop out. And his activities were really gained an awful lot of public and media attention. And it was at a time in the United States where there was a massive polarization around the politics of the Vietnam War. And arguably, Bob can talk about this, but arguably some of the fissures that developed in American society at that point are still echoing through with us today. And the side that was against the war, predominantly young people, predominantly people at risk of the draft, but particularly well-educated college people, were the ones who took these drugs as a recreational thing, and a subgroup of them saw them as simply a different way of living, a different way of seeing the world, an alternative culture. And this really produced, because of its alignment with the politics of the time, produced a kind of moral panic in the US government, which soon after this sort of became obvious as a problem, developed the notion of the war on drugs. And the government response to this was really the abandonment of the project by serious scientists. And there were a number of reasons for that. One is that the evangelism of these people was unattractive to, if you like, a serious group of relatively conservative people who were the scientific community. So the evangelism was unattractive, and it was felt to be anti-science in many ways. The demedicalization of the activity, the dominance of this recreational model, really made it seem improbable that it could be used clinically. And there were undoubtedly bad outcomes for people using these drugs under unsupervised conditions. And so the upshot of that was that the use of LSD and the related drugs was made illegal in about 1970. But interest in research had already tailed off before that. And I think the last NIMH grant was several years before the drugs were actually finally put under this with the likes of peyote, ecstasy, lysergic acid diethylamide, which is LSD, and indeed all of this class of drugs were placed into this schedule, which has no accepted medical use, with a lack of accepted safety and high abuse potential. Medications within this schedule may not be prescribed, dispensed, or administered for medical use. So that, if you like, was that for research by responsible clinicians. And the only people who could use these drugs effectively were breaking the law, and they were happy, therefore, to be underground in using them. And the underground era lasted really through to the present day, and it still exists in the United States, really particularly famously on the West Coast, but actually in most states in the US, there are people who call themselves underground guides for people who are seeking psychedelic experiences. There was also innovation, chemical innovation. In this guy, Alexander Shulgin, was a chemist for the Dow Chemical Corporation. He, in fact, invented the first biodegradable pesticide, and he was rewarded for that by basically complete independence. I mean, he was given a free role within the company to just do what he wanted. And he then, and also a substantial amount of money to live on. And so he set up and is credited with the discovery and synthesis of over 200 psychoactive compounds. And he innovated around the basic LSD type ring structure that you saw earlier. If you want to read about what he did, then this book, PCAL, he wrote with his wife, is about his life and how he got into and investigated these drugs, because he wasn't content with just synthesizing them. He took them, all of them, tried them, usually with small groups of friends, and he invented his own way of describing them. And if you want to see the way he lived and the rather chaotic state of his laboratory, then this film, Dirty Pictures, is available on Netflix and is quite entertaining. But he was a big character, Shulgin, and he has obviously created a lot of chemical entities which have the potential to be investigated if it turns out that this class of drug is useful for psychiatric treatments. Now, I think we need to go back. So the underground era continued really through to, as you can see, I've used as a rather arbitrary date, February 2014. But around that time, and up in the 2000s, there had been a recrudescence of interest in investigating these drugs, first and foremost in healthy volunteers, and then the idea arose that the original description for a use in psychiatry might also be looked at in a little more detail. And one person who had all along been calling for this was a guy called Rick Doblin. He is ran the Multidisciplinary Association for Psychedelic Studies, and this was a project, particularly the highest priority project, was sponsoring FDA drug development research into MDMA-assisted therapy. And Rick Doblin is shown on the left here, and this is his role as a CEO, and this is his role on the lower picture, perhaps the way you would like to be best remembered as a man interested in psychedelics before his time, sitting with Hoffman, who was, of course, the discoverer of LSD. So what MAPS over 30 years has aimed to do was to try and bring back the use of psychedelics into specifically psychotherapy. So this is a model which is essentially psycholytic, if you can remember the old terminology. And the idea is that psycholytic treatment, particularly MDMA, will allow patients to use this psychotherapy that is used anyway to treat, for example, PTSD, in a more effective way to achieve more profound outcomes, to perhaps attain them quicker. And their first target has been post-traumatic stress disorder. And they have gone as far as is possible through, because they got breakthrough therapy designation from the FDA, they are now well along a development pathway into so-called phase 3, which should allow them to take a dossier of clinical trial data to the FDA for approval for this particular indication. The data is based on this trial design. So this is MDA-assisted therapy for severe PTSD. This is a phase 3 study. Perhaps I'll just move that if you can see. And what you see in different colors are the different kinds of interaction. So there's a screening, there's then assessment, there's preparation, there's tapering of other medications, another preparation, more tapering, and then finally, as you can see, an experimental session that takes place after various kinds of enrollment and, after various kinds of measurement and ratings. And the patients have 3 of these experimental sessions, and between each there are integration sessions that allow patients to reflect on the time they've had under the experience of the medication, and to integrate that into their thinking about their symptoms and their illness. As you can see, overall, the study lasted about 18 weeks, and the total number of sessions are 3 preparatory sessions, 3 experimental sessions, 9 integration sessions, and 4 endpoint assessments. So there's a lot of contact here. So there's great deal potential for what one might call a placebo effect. And the key thing is that the actual experimental sessions, patients are randomized either to receive drug or placebo. And the difficulty with this is that the amount of psychological treatment layered into this, the extent to which there's expectation bias, and also the problem that blinding is virtually impossible if one gives large doses of these kinds of drugs, makes it tricky sometimes to interpret findings. But nevertheless, findings are what we want in order to understand the, what have I done here? Yeah. So this shows you the results summarized on this slide for this phase 3 clinical trial. And the summary is shown on the left is the outcomes after 1, 2, or 3 sessions. Each column refers to those outcomes for a placebo plus therapy, and on the right, the outcomes for MDMA-assisted therapy. And what you can probably see if you look particularly at the third column is that the number of patients who are described as losing a diagnosis or being responders runs at somewhere between 50 and 30% in the placebo group. Whereas by contrast, you can see that in the patients who were treated with MDMA, you have a much larger number of patients who lose the diagnosis, and up to 60% if you include people who are responders to the treatment. So this has been regarded as very promising. It's still quite a small study, even though it's described as phase 3. And I suppose the other thing that one has to say is that the outcome here is based on a scale called the CAPS, and it's also based on the effect, the impact on a single outcome, the most significant post-traumatic memory that the patients experience. And the difficulty with that is that it maybe risks isolating one element of the total clinical picture. And these patients often had quite complicated PTSD, and therefore were quite distressed. And therefore, we can't be completely sure that the overall quality of the response is as good in total as it may be for individual symptoms. So with that caveat, I think I'll leave this, but there are also other concerns about this trial which have surfaced recently and are no doubt going to have to be worked out because of the misbehavior by at least one of the therapists who appears to have been involved in the study. So this will work itself out in due course, but it's going to make things more complicated than it would otherwise be. And because this is essentially described as a psychotherapy, it's already going to be complicated for the FDA to consider this kind of data. Now, I've described that as psycholytic therapy because the doses are modest and patients remain in contact enough with reality to talk to have a dialogue with the therapist, and in fact, usually with two therapists. The new psychedelic therapy goes back to that model of a transcendent experience on a high dose of one of these drugs. And the people most associated with developing this on the left is Roland Griffiths at Johns Hopkins University. And then on the right, Robin Carhart-Harris, David Nutt, and Amanda Fielding, who is a long-term campaigner for the availability of drugs for medical and other purposes. And they in the UK did studies at Imperial College, which have had a big impact in the field in recent years. The first of these studies, which had the impact I describe, was the use of psilocybin to treat depression and anxiety in cancer patients. And this was done by Roland Griffiths and colleagues at Johns Hopkins Medical School. They enrolled 51 patients with end-stage cancer. These patients were essentially had the existential distress, but along with that, a lot of anxiety and depressive symptoms. And these patients were put into a double-blind crossover trial design where they received either a low psilocybin dose, which was either 1 or 3 milligrams per 70 kilogram, to a high psilocybin dose, which was 22 or 30. These doses were changed during the course of the trial. But basically, this is a low or placebo dose, and this is a high and clearly psychedelic dose. And what you can see is that if you look at over on the right here, in the group who this is rating depression, the people who had the low dose first had something of a placebo response. So something like a 20% response, which is not uncommon for depression trials, of course. If those who had the high dose had something like a 90% clinical response. And those patients who actually showed remission were similarly running at very high levels for the high dose arm of the study, something like 60%. And these effects were extremely rapid, so that patients really responded virtually from the day on which the dose was delivered. And there was an effect also very prominently on anxiety. So again, if you define response, that's a 50% reduction in symptoms. You're looking at something like 80% versus 20%. And if you look at remission, and bear in mind that remission is a very high bar, you see patients who essentially return to normal mood in 50% of cases after the high dose. And the patients in the low dose subsequently responded when they received the high dose. So these effects were very striking. They were obviously in a specific patient group. There was this complication of the cancer, and there was also the complication of the crossover design, and the difficulties of blinding, which I've already described. And that's difficult both because the patients know, the doctors know, and if one's not careful, the raters who are looking at the patient's outcomes know. So it's a problem that's very hard to get over with this approach to treatment. Now, what was the mechanism? I'm just going to show you here that They used as a measure what they call a total mystical experience score. And you can probably see that in the two cases, so these are the when patients are responding either usually to the first or the second high dose, you can see there's an association between the total mystical experience score and the change both in the anxiety and the depression score, slightly higher in fact for the anxiety, but these are both statistically significant. And in addition, these total mystical experiences scores were very highly meaningful to the patient. So again, this connection with a quasi-religious experience is encouraged by the use of a scale that describes itself as mystical experience. In fact, what is very interesting about this, and the Imperial group who've gone in and studied this in more detail, they suggest that it's really the what is sometimes called the connectedness, the oceanic boundlessness that is produced as an experience by high-dose psychedelics that is both strongly associated with good outcomes in depression and anxiety. And that is very interesting, and it kind of relates to what Bob Cloninger was talking about in terms of restoring a sense of connectedness with the world, which of course can be related to meaning and can be religious. But it doesn't really have to be. It can be quite a secular experience as well. But it's a very important one to patients who become depressed and hopeless, particularly in the context of a fatal disease, which these patients had. Now, what I'd like to talk about further is just how we're going to remedicalize these conditions if we think it's worth it. The Hopkins group saw the existential distress of patients with cancer as a kind of obvious way to use drugs meaningfully. And they were very influenced by the idea that it was a sort of spiritual experience. The problem with that is that there isn't actually, in our modern diagnostic terminology, and certainly not for the FDA, there isn't such a thing as existential distress, which forms a target. It isn't kind of in DSM. And they hadn't really diagnosed patients as having major depression, etc., although they clearly had got the symptoms. And major depression isn't a very high bar anyway, as you'll be aware. But the route to remedicalization really requires that one has a conventional regulatory target. And so what is now happening is that there is an effort to try and, following, again, exploratory trials by academics, is to try and discover whether these medicines are useful for treatment-resistant depression, as you've heard, for post-traumatic stress disorder. There's also interest in substance use disorders and obsessive-compulsive disorders. And then finally, of course, treatment-resistant depression is just major depression that hasn't responded to other treatments. So there's some interest also in major depression. And there's a number of different groups who are taking different routes to try and produce the necessary data that will allow the FDA to approve these drugs for use in man on the basis of their efficacy and safety in a known condition. Now, the reason that I'm now Chief Medical Officer of a company is I'm interested in this project, and I joined this company, Compass Pathways, in the middle of last year to try and facilitate the move from a phase 2 study, which was just about to read out, to phase 3, which we hope may be able to start towards the end of this year. And the reason that treatment-resistant depression is important is that it's so common. It's estimated that certainly about a third of patients who present with major depression really fail to respond to first or second line treatment. And you'll recall the data from the STAR-D that suggested that as well. And that means that when you get to third line treatment, you've got a real problem because response rates are really quite low, and there's quite a lot of patients who are left in a demoralized and in many ways a worse state than the average person who has depression that's treatable. So this is the unmet need, and it means that there's both a medical and a commercial reason for trying to develop something that works in this patient group. The particular possibility of doing this and having it paid for by commercial investment is that there is a patent which is developed by the founder of this company for psilocybin as a synthetic high purity polymorphic crystal, which is called COMP360. Now, this is psilocybin, but it has a particular patent. It has, because of the way it's prepared and because of its stability, it has a patent protection that is potentially allows this to be developed, and that gives a certain moat so that there is freedom to actually do the necessary work and have some patent protection over a number of years, so that the money that is spent on developing the medication can be recouped over the subsequent 6 to 11 years, depending on where you are. So we have the drug, and that is the we think the primary ingredient, but we also know that we require psychological support. And we think that psychological support we should provide is essentially a safety measure. We don't really know how to facilitate the actions, the pharmacological actions of these drugs, but we do know that people need to be prepared for the experience they're going to have. So the psychological support means preparatory meetings with someone who can explain to patients exactly what's going to happen. It's the use of a digital platform that allows them to access more of that information when they want, so that they can, in a sense, complete a course, a psychoeducational course on the use of psychedelics. And that is combined with the day of treatment, before the day of treatment, and on the day of treatment. Again, you need a therapist to be with you the whole time to support you and to make sure things don't go wrong. This approach which I've described on here, was designated as having breakthrough status by the FDA, just as the PTSD application for MDMA was. And there was a phase 1 completion and then phase 2B clinical trial, which was recently completed. And this is the design of the trial for those of you who like clinical trials, which I certainly do. So these were patients with treatment-resistant depression. Over 90% of them were naive to psychedelics. So these were patients who came through conventional pathways. That hadn't been as true as some of the previous studies that had been done in academic centers. These patients met the criteria for treatment resistance because they had failed at least 2 and no more than 4 treatments of a conventional kind for their depressive episode. The depressive episode was no longer than 2 years, so we didn't want people with too chronic. We wanted a realistic kind of clinical population. And patients had to withdraw from most of the psychotropic medications that they might be taking, and in particular, the SSRIs. And that meant that we had to allow quite a long delay, potentially, both to prepare patients, but also to withdraw them from SSRIs. And that proved to be highly feasible. In fact, when patients are well motivated, the problems with withdrawal of SSRIs are rather minor. So after this preparatory phase, patients were randomized to receive either 1 milligram, 10 milligrams, or 25 milligrams of psilocybin. And that was administered on a single day. And the patients remained in the session for from 6 to 8 hours. They then went home and they came back the next day, basically to complete an assessment, formal assessment. And there was repetition at weeks 1, week 2, week 3, and week 12. So we tried to continue this out to 12 weeks, although of course we knew that at some point patients who had not responded or had been in the low dose group might want to go back onto treatment. There were also a couple of remote assessments at weeks 6 and 9 to stay in touch with the patient. Now, the primary endpoint was the reduction in symptoms using the MADRS rating scale at 3 weeks. And the MADRS in all cases was administered remotely by blinded raters who did not really know the nature of the study, let alone any of the details about the patient. And that's the main protection, we think, against bias in this sort of clinical trial. And what were the findings? Well, what this shows is that there is a dose-effect relationship So in blue, you can see the 25 milligram dose and its effect on the MADRS scores. So the reduction, as you can see, of about 15 on the MADRS for the 25. And this is from, a baseline MADRS of around 30 to 33, which is basically most of the patients were severe according to MADRS definitions. But moderate to severe would be the group that are really being studied here. So you can see 25 milligrams, 10 milligrams, and 1 milligram curves. The red is the 10 milligram, the dark color here is the 1. And you can see that there is an immediate effect, which you can measure at day 2. Many patients in remission on day 2, just as I said before, it's an extraordinary effect, with some loss of effect as we observe patients up to 6 and then up to 12 weeks. But a more marked loss of effect in the other patient groups who are drifting up through over that time. There's a highly statistically significant difference of 6 points at 3 weeks. And of course, that's compares very favorably with the kind of differences that are described in trials with SSRIs, which are sometimes less than 3 points on the MADRS. This is, of course, all very well, but the question is how many of these patients don't actually show this drift and remain well? And that is interesting because there is a significant fraction of them. So the numbers of patients who were sustained responders out at week 12, so that is patients who'd not received any further treatment, they'd had a single dose of the psilocybin, about a quarter of the patients in the 25 milligram group remained in that state at 12 weeks. So a certain subset of patients, this experience has quite a kind of, if you like, it kind of throws a switch. It changes the way they see things, and they really do show a very sustained and durable response. But it leaves us with a lot of people who do not have that. And obviously that's going to be something that has to be investigated. Can we change, improve this number? Is this just a characteristic of the TRD population? How can we get these numbers better? But the patients who do respond have a very high quality response. And I want to illustrate that just by looking at their ratings of their own clinical condition. So I'm comparing here in blue the good responders that I've just shown you with non-responders. These are people who may have a psychedelic experience, but they have no clinical response. And what you can see is that the self-rating of depressive symptoms falls in the well-treated patients. So this is selected groups. They're illustrative. It's not a simple. It's not. You can't give statistics to this. This is just illustrative. But what you can see here is that the levels on the QIDS, which is a self-rating depression scale, fall to under 5, which is basically what healthy people score themselves as when you use the QIDS in them. And the quality of life scale goes up from 0.5 up to somewhere between 0.9 and 1, which again is where a healthy population scores itself. And then finally, we see a sustained increase, at least for the first 3 weeks, in positive affect, which again is something we think may be a particularly useful property of this approach to treatment. So the quality of the response in these people who do respond is very, very good, and it's well sustained. Now, the flip side of all of this is how tolerable are the adverse effects? You know, how much are they? Are they a high price to pay for the treatment outcomes? And basically, this just shows you the summary that obviously for some of them, like nausea and fatigue, are related to the dose of the drug and are all recorded immediately after the treatment. So they're recorded on the day following the day of administration of the drug. And patients are asked what they complain of in that time, any adverse effects. And these are the kinds of things they describe. So most of the effects that are described are not severe, and they occur immediately on the day of, and they're sort of to be expected. And what we need to work out is which of them are really wanted, which are a part of the treatment, which are the unwanted and are anti-treatment, as it were. And we still have to work on that. So effectively, what is very reassuring is that this is a well-tolerated acute experience for patients with treatment-resistant depression. And we couldn't have known that before these studies were done. So that's very reassuring. However, as always in depression trials, suicidality is a big issue. And suicidality wasn't a feature of the first day, but we did have people who became suicidal over the course of the follow-up. And you can see the rates at which that occurred was something between 5 and 7%. And those rates are what you probably would expect by comparison with other trials which have included treatment-resistant depression patients. So you have to accept that this is an issue for this approach to treatment, that we're going to have to, as with all treatments for depression, be vigilant about the clinical course. But on average, there were no differences between the three arms. There were some serious adverse events which occurred in the 25 milligram arm, and we can't explain why they clustered in that arm. There were only 3 important ones. And we obviously keep an eye on serious adverse events, which are classified rather strictly in relation to suicidality in a way that slightly amplifies the true risks of the usual events that are being captured. But that's a technical matter, but it doesn't diminish the fact that we'll have to keep an eye on suicidality as an issue for patients treated with this modality in the future. These patients were all withdrawn from SSRIs. Could it be that psilocybin could be adjunctive? Well, this is a study that's actually done in healthy volunteers, and it shows that if you put healthy volunteers on escitalopram or placebo and then give them a high dose of psilocybin, then in fact, the co-treatment with the antidepressant SSRI doesn't actually affect the magnitude of the psychedelic experience. So this is our preferred measure, which is called the Altered States of Consciousness Scale, which has 5 dimensions. And what you can see here is that basically the placebo and the escitalopram numbers cluster very closely for all of these different measures, which are described along here. And the only one that separates a bit is, in fact, an anxiety dimension, and it's actually rather less in patients treated with escitalopram. So this opens the possibility that we can use psilocybin as an adjunctive treatment with SSRIs, as well as in patients who have withdrawn from SSRIs. That wasn't known before. We have very preliminary data that supports this interpretation, and we shall see whether that turns out to offer a better choice to patients in the future. So that's where we are. I've given you a history of these drugs. I've told you a little bit about the original medical era, how it all went wrong, really, and how it, I think, could go wrong again, and how it's now going much better. And I think we're at a stage now where we may be able to embark on a phase 3 program, which over several years may allow us to bring this approach to treatment to patients in clinics across the world, hopefully. There is a political dimension to this, and I think it's worth understanding that where we are. The use of the compound is I've tried to show on the top here. So there is simple illegal use, whatever that's usually, essentially, the way we're positioned we're in at the moment in most countries. There is really no regulation. There's simply none or law enforcement. And the leadership, the provision, is really by people who are prepared to break the law, which I think is a problem. There is a ritual or traditional use, and obviously we're the WPA, we've got to take account of the whole world, and there are many societies in which there is a ritual and traditional use of drugs of this kind. They're usually related to various forms of belief system, and the people who run them have the status of priests, although in some cultures they're called shamans or whatever. But obviously this is one of the ways in which these drugs are used, which we have to respect and is open for people to develop. But in the West, it doesn't seem very likely that's a route we would logically take. We don't have a structure to pin that onto. I think it's much more sensible to think of our culture as being better adapted to a medical approach. And I think in most cultures, actually, a medical approach is probably going to be the safest. If you take a medical approach, then you have to use the conventional models for deciding whether drugs are efficacious and safe. And that means very expensive development programs. And the only way to do that, I think, is to have private investment that allows people to make a profit eventually from the development of the treatments. But the key thing about a regulatory model is that the cost to patients who can't afford it can be reimbursed. And I think it should be psychiatrists who develop this approach. Finally, if we say that these drugs should be freely available, they should be decriminalized, made freely, make it no penalty to possess them, then essentially they become recreational, and the leader's basically going to be Hollywood. And I don't think that's, I think that in that direction is potentially a disaster for various reasons. So I think that the route to fair and safe access is regulation, not recreation, and that remedicalization is the best route to legalization. And that's the reason I'm Chief Medical Officer of a private company. So thank you very much for your attention. I'll be very happy to take questions.
Thank you, Guy, for the extremely modern topic and the detailed presentation. We don't have any questions. I just wanted to, we don't have questions from the audience, I mean. I just wanted to tell my experience with ketamine, which was unexpectedly positive. Almost all patients we enrolled in ketamine, and they were super refractory. Our patients are super refractory. When we say that someone is not responsible, it's not because he had 2 trials of low dosage, middle dosage antidepressants. They are super refractory, and most of them, almost all of them, did very well, exceptionally well on ketamine. We will see what will happen on the long term, but in the short term, the results are impressive. We do have one question I see now. Do you think ketamine effects on treatment-resistant depression can be explained by changing the mind of patient rather than just a pharmacodynamic effect?
We're very interested. I mean, it's a very interesting question how these drugs work. As you will know, ketamine has really been developed as a drug, and there's in most centers very little emphasis has been placed on the actual experience of the patient. We also know that when you give these drugs to animals, you have fairly consistent effects on emotional learning and also memory. I'm very much mindful of the fact that Bob was making the argument that we should think about underlying mechanisms in relation to personality. And obviously that's the common substrate for thinking about our treatments as well. And what is very interesting is that if you look at the emerging literature on ketamine and related NMDA acting drugs, there is a very immediate effect on memory. There isn't so much of an effect, and that I think is the basis for this very rapid antidepressant effect. There is less of an effect on new learning. And what is quite interesting is that if you look at particularly psilocybin, where we have most data, there is an effect on memory. So you get the same kind of effect as you get with ketamine, but you also get an effect on new learning, which is actually a little like what you get with the SSRIs. And of course, the SSRIs take quite a long time to work, and that's almost certainly due to the fact that you have to learn new associations under a different set of reward contingencies and biases. So I think that we know that as far as the neural basis goes, then there is that kind of common substrate. What, of course, people are very fascinated by is that when people have these psychedelic experiences, they describe existential changes, that they have insights, they have different perspectives, which they attribute purely to the event on the day you take the drug. And that, of course, is what is still very fascinating about this. Is that really what matters? or is it the effect, if you like, on the serotonergic system, which is also quite profound and lasting, that makes the difference? And indeed, how do we optimize the interplay between the two? So I don't know whether that really answers the question that was posed, but that's the way we're thinking about it. It's a very interesting and exciting area to get into at the moment, I think.
We have a second question. Any thoughts about the combination of psychedelics and virtual reality as a therapeutic approach for psychiatric disorders?
I think, yeah, I mean, the two aspects to that I think we can probably prepare patients for their psychedelic experience using virtual reality or virtual unreality, really, so that they are sort of better prepared for what's going to happen. So that's one use of virtual reality. The second is that, you know, virtual reality can be used in various kinds of exposure treatment. And what will certainly happen in the future, I think, is that patients will receive a psychedelic and then they will have psychotherapy subsequent to the integration. The integration really just deals with the experiences on the day, and they can be very frightening. It's like people take this, you know, the trip analogy is quite a good one. People encounter, you know, nightmarish things along the way, and they need to sort of sort that out in their own mind. But there's quite a lot of evidence from the animal work that they're then they're in a more, their attention, their flexibility, their if you like, their plasticity is increased. And this is what actually people describe, is that they find habits easier to break. And that's obviously good if they're bad habits, not so good if they're good habits. But they're almost certainly more susceptible and more sensitive to psychological interventions. So if you think of psychotherapy as essentially relearning, as a learning process, if you're facilitating emotional learning, then it should make other specific psychotherapies more effective. So logically, you would actually use something like CBT in the phase after you've had the psychedelic experience to capitalize on the months that seem to leave you with this enhanced sensibility, where one is more open. I mean, the way people describe it, they feel more open, they feel more connected, they feel more meaning in things. And there's obviously, that's a time when there's great potential for change, which I think our structured psychotherapies can offer something quite useful.
There are two more questions from Roland Ivanovs. Are there any markers for responders and non-responders? And where there are any training opportunities for this method?
Yeah, the marker question is a very good one. I mean, we don't yet have a marker for people who are not going to respond to treatment. You know, that is, we can't select people in advance and say, this is an enriched population, except I think probably MDD is going to be a more responsive population than TRD. I mean, that's very likely because we've chosen difficult to treat people in this. So in that sense, we can't choose people on the basis of existing biomarkers. We do get a clue as to whether people are going to respond, however, from, as I think I showed, in that the intensity of the experience, particularly of being connected to the world on the day in which you're dosed, is a predictor of outcome in some of the studies that have been published. So in a way, you get an immediate feedback as to whether your dose has had the effect that you want. And you know that if you have the high dose effect that you're looking for, particularly on this sense of connectedness, then you are more likely to get a treatment response. So that's a kind of biomarker which will be useful clinically. Training, well, we're setting up a big training program for the clinical trials that we're going to make up the phase 3 program. But of course, these medications are not yet available except under the underground. So this is going to be a few years before clinicians are going to get their hands on things. But, you know, we're looking forward to the time when they do and they can start using these medications appropriately.
So before I give the floor to Afzal, I would ask Robert, who is waiting in the background, if you have any comments, Robert.
But I think fascinating talk to really enjoy this. The puzzle is, it's still hard to know what's causing this sense of connectedness and positive outlook. And clearly, the key to the self-transcendent experience is the perception of unity and being a part of something boundless and that you're not separate from. And so it's breaking that idea of separateness that causes so much pain to people. And so your question, I think, about is this a pharmacological effect mediated through serotonin? Is it some interpretation of that then is overlaid by some people? And why some people and not others? It's almost like for a short time, you're reawakening their youth that is somehow, but I wonder if that could be tested by looking at changes in transcriptome. And during that period where there's new learning potential. I'm just speculating because there are dangers to the unsupervised use. I was actually working at Berkeley in 1969, in the summer of love. And a number of people that I knew using drugs unsupervised, when I'd come back from my research, committed suicide on bad trips. And so I saw the sickness that came from it. And it makes me concerned that at the same time, I'd see the possible potential that you may be opening a door again, at least for a time.
Yeah.
And the question is how we can proceed to understand that mechanism.
There's a very brilliant woman called Gull Dolan who's looked at what are essentially critical period behaviors in animals. And she's shown that giving psilocybin actually restores social learning that you only normally see in very young animals, and you get it in older animals. So, I mean, I think, there are a number of really interesting leads here. And she also does some of the underlying gene expression. So, I mean, there's clearly implications for mTOR and a number of the systems that are kind of great interest to aging and to synaptic plasticity, et cetera, et cetera. So, I think the story is only just starting to be written, actually, of just where the potential begins and ends for these meds. And of course, the sadness is that, from 1969, we did nothing with them really that was of interest. And now, of course, there's new interest, and I hope we'll be able to fund the research that's needed to find more out. And I hope that governments will step up and spend more, but for clinical trials, it's just become, as you know, very, very expensive.
If I may add a detail, it is the rule rather than the exception that any medication which is beneficial for patients could have a deleterious effect on healthy humans. For example, if you give insulin to the diabetic patient, you save him his life, and also you protect the brain. But if you give insulin to healthy individuals, eventually you produce brain atrophy, cortisone, antibiotics, almost any medication that saves lives when given in healthy persons will cause some kind of damage. So the key issue here is that a side effect is the euphoric or the whatever existential desirable effect can be useful for recreational effect. So Robert, what you say that it could cause suicidality. Yes, anything can cause harm in the general population. The problem is the effect that promotes recreational use, which I don't think we will be able to overcome unless we have decode the true way of the therapeutic effect is carried out, and then we synthesize agents that possess only this and not the rest of effects, which I'm not sure. I'm not sure we can disengage these two effects.
Well, that's a big question. I mean,
we don't know.
That's what. I mean, there's a number of companies who are chasing that very question. Whether the effect, you can get an effect on the serotonergic system that gives you, a potent antidepressant without having the experiential experience, without having the experience that we think is also potentially helpful to people. And there's some people who argue we should be giving it under anesthetic. That there's no necessity for the risks associated with the actual experience. But I don't know. I mean, we're going to see whether we're at the very early stage of serious investigation, and it's going to be interesting to see where we get to. But one thing's for sure, we've always known they had powerful effects on the brain, and I think we kind of just as Bob's saying, I mean, unsupervised, these drugs have a great potential to do harm. I think supervised, they have a corresponding potential to do quite a lot of good, but we need to establish what that is, for whom, how durable the effects are. But people, even the general population, the commonest experience when the drug is taken under supervision is a very positive one. And, about 30% of people say it's the most positive thing that's ever happened to them. So, even in the general population, there is a perceived benefit. And it's to do with this sense of being connected and a life being meaningful and so on, which is intriguing.
Yes, Robert, yes.
Is there evidence of any impairment of declarative learning for a while after the sessions?
Well, the Compass did a healthy volunteer study, which is published in the Journal of Psychopharmacology, and they did quite a bit of fairly standard CANTAB, neuropsychological type testing, and they didn't detect any differences there. Obviously, you might want to ask a specific question that isn't answered by the test battery that they used, but certainly there aren't global impairments of attention, short-term memory, new learning, episodic. None of those things are impaired for sure. But of course, you might be interested in autobiographical memory or, right, as you say, procedural stuff as well. So there's a lot we don't know.
Now, the question is whether you're enhancing the ability to use self-awareness or default mode versus impairing or shutting down temporarily the analytical problems that are sometimes, especially with the anxiety and depressing disorders, problematic, being overly self-critical.
Yeah, well, I mean, certainly the fMRI studies are very interesting because what they show is that it weakens the default mode network and you get a much richer set of connections between different brain areas that are not normally very highly correlated. Of course, that's the effect of that's the acute effect of the drug while the drug is on board. And of course, that's very striking, and it's not surprising given what's going on in people's minds. You would expect that would be what happens. But of course, it's quite interesting. The default mode network often looks stronger in patients with particular disorders like anorexia, OCD, depression, where being stuck in a way of thinking is a very much a feature of the psychopathology. And where, you might imagine, it's a very attractive idea without real substance yet, But the idea that you could weaken that sort of pathologically strong default mode network with the drug is kind of what's also one way of thinking about it, which is kind of more brain-based than psychologically based.
Okay.
Afzal, Peter?
Let me congratulate Professor Guy Goodwin for a very brilliant presentation. This reminds me about his discussions, scholarly discussions, when he was in the MRC Psych unit in Edinburgh, and I was a trainee over there. So it really reminded me the way he used to teach us, the way he used to present in our journal club. So thank you very much for a very, very clear presentation, Professor Goodwin.
Thanks for taking me back.
Just one suggestion. Because this is going to be an important issue in psychopharmacological domains of psychiatry in the coming years. Is there any chance, if through the help of Professor Fonteclas, especially with his WPA section on evidence-based psychiatry, if we could have some discussion document giving some practical advice about psychedelic drugs, their side effects, their uses, their contraindications, because WPA gets a lot many requests for some small synopsis, short descriptions, which can be used, especially for our colleagues in low and middle income countries.
Yeah.
Yes, Professor Fonteclas, would you like to
We
support this?
could initiate some kind of a guidance paper. I wouldn't see a monograph. I wouldn't see a small monograph at this as a starting point, but a guidance paper would be. I think we could do that.
Because this is going to stay as an important area in psychopharmacology.
If Guy finds this a good idea, we can surely try to.
We can explore this later on with him.
Yeah.
Well, I think you're right. It's going to be important, but it's going to be a long time before these drugs are approved for use. We're right at the beginning. That's a phase 2 trial. We've not even published the data I showed you tonight yet. So.
Guy, what is the problem? Maybe not a problem per se, but still it's a concern that now that we know that certain stimulants like ketamine are useful in the treatment of depression, especially in the US, but also in Europe, we will find a lot of desperate patients with depression and maybe with other mental disorders trying to find anything in the black market, in the streets. One thing is what research says about medication use, treatment use of a specific kind of substances, and then we need some kind of guidance of how to treat and how to manage all those people that will go out and imagine that at some time we might find a significant number of patients self-medicated by even ever higher dosages of new and very novel and uncharted substances.
Yeah, no, that's absolutely, you're 100% right. And some of these substances you can go and pick in your garden, so it's not like we can regulate them anyway. So it's, I think you're onto something, but it's sort of, you kind of need quite specialist knowledge actually about these unconventional alternative sources. It's not. It's not traditional psychopharmacology in a way. It's, what you need to be careful about in terms of what you buy. And then, of course, you don't actually know what you're buying. That's the other problem. So it's a tricky area. I mean, I think I can clearly see the need for some sort of simple informational level communication. And it would include ketamine, of course, because ketamine is now becoming very widely available as a treatment without any, because it is a regulated product, so it can be obtained legally. It can be used off-label, really, without. So I think certainly ketamine is a starting point, and then these plant-based things are also. You need someone who knows about them because in a sense they're not. It's quite a specialist area.
So Afzal and Peter, you have the final words.
Well, I think we should really thank our two distinguished speakers for not only giving excellent presentation, but starting a very, very informative discussions. And we are grateful to you, to your team, for continuing with these educational seminars, educational activities. And we hope that, you look at the number of people who are attending on the Sunday evening. This is amazing. And this shows the expertise of our speakers, that they have attracted so many people to come and listen to them on Sunday evening. And we hope that we will bring them again to the WPA meetings and the congresses, and we will continue using their expertise in the future. So thanks again, and thank you very much for that.
Thank you, everybody, for being here with us. Take care until next time.
Indeed.
Bye-bye.
Cheers. Cheers, Bob.
Thank you.